Okay, so welcome to this next video in which we are discussing protein folding and protein folding diseases. Okay, so in the previous video what we looked at was the ubiquitin proteasome pathway, which is one of these pathways by which misfolded proteins are degraded by the cell's machinery. Okay, so we saw how you first tag the condemned protein by producing this polyubiquitin tail that's attached onto the protein, and then this polyubiquitin tail is recognized by the 19S regulatory particles of a 26S proteasome, and this results in the condemned protein being fed through the 20S core of the 26S proteasome and being degraded as it's fed through that 20S core. Okay, right. What we now want to turn our attention to is we want to finish the proteostasis pathways. Okay, so I just want to talk a little bit about autophagy, okay, which is the other major degradation pathway within cells. And then what we're going to move on to is the protein folding diseases, and we're going to start off with the loss of function protein folding diseases. Okay, right. So firstly, let's talk about autophagy. Okay, autophagy or autophagy, however you want to pronounce it, literally means self-eating. Okay, phage means in some old language, I presume Latin, eat. Okay, and auto means self, so this means eating yourself. Okay, so this is basically like phagocytosis. Okay, which is something that occurs within certain immune cells, in which the immune cell basically eats pathogens, but instead of destroying pathogens, what we're going to be doing is breaking down our own cells' components. Now, there are different forms of autophagy, okay, but they all involve the lysosomes. So let me just explain what lysosomes are. So if I draw a picture of the cell here, okay, here is the nucleus. Okay, lysosomes are small little vesicles that are in the cytoplasm of the cell. So we'll let these represent the lysosomes. Okay, and the lysosomes contain enzymes which are capable of breaking proteins down. Okay, so these enzymes are specifically called hydrolases. Okay, and hydrolases are enzymes which are capable of breaking bonds using water. Okay, so they're capable of hydrolysis reactions. Okay, and there are actually greater than 50 different hydrolases within the lysosomes, and the lysosomes are going to be very important in a moment when we discuss the lysosomal storage diseases. Okay, so greater than 50 hydrolases. Okay, right, so these hydrolases are capable of breaking down proteins. So autophagy basically means using these hydrolases that are stored within these lysosomes to break down proteins. Now, there are many different pathways by which you can actually recruit these hydrolases stored within the lysosomes. Okay, and we're just going to look at two. Okay, the first one that we're going to look at is the one that is used to destroy single proteins. Okay, and this is what's known as chaperone-mediated autophagy. Okay, and for short, chaperone-mediated autophagy can be abbreviated down to CMA. Okay, so C for chaperone, M for mediated, and then A for autophagy. Now, what this involves is a particular protein, a particular chaperone protein, binding to a protein that is destined to be destroyed by the lysosome, taking it to the lysosome and then delivering it across the membrane of the lysosome into the lumen of the lysosome where it will be destroyed. Okay, so a specific example of a protein that is very involved in chaperone-mediated autophagy is the protein HSC 70, okay, and this stands for heat shock cognate protein of 70 kilodaltons. Okay, so this is the heat shock cognate protein. Okay, and that's what the C is for, cognate, and then protein, and then it's of 70 kilodaltons as before. Okay, now the function of this protein, HSC70 then, is to bind to proteins that are contend to be destroyed within the lysosome. Okay, and I'll show this here. So let's say this is our HSC70 protein here, which I'll colour code in blue. It can bind to proteins that are destined to be destroyed by the lysosome, and it delivers them to the lysosome. It then interacts with a complex on the membrane of the lysosome to deliver the protein into the lumen of the lysosome. So here is the protein that it's binding to there. Okay, so that's one way of destroying proteins, by sending them to the lysosomes to be destroyed. Okay, now there's another form of autophagy that I want to talk about, which is what's known as macroautophagy. Okay, and it's this form which can be used to destroy whole aggregates of proteins which have formed within the cytoplasm of a cell. Okay, so let's say that we have a protein aggregate here in purple. So let's say this massive great purple blob that I'm drawing here represents a protein aggregate, so loads of misfolded proteins that have aggregated together. Okay, the macroautophagy pathway is a means by which we can destroy this aggregate. Okay, so we can't get this into a lysosome, that's too difficult. So what we're going to do is we're going to sequester it off from the rest of the cytoplasm, and then we're going to fuse a lysosome onto that separate little compartment that we build. So effectively what we're going to do is produce a vesicle around this and then fuse that vesicle with a lysosome, and then the lysosome or more contents can be emptied on top of this protein aggregate. Okay, we just firstly need to separate it off from the cytoplasm. Okay, so what happens is you bring a vesicle like so. So this is a vesicle, a normal vesicle, and what you're going to do is you're going to bend this vesicle around your protein aggregate to make a membrane around the protein aggregate. And this is what's known as an isolation membrane that is curving around this protein aggregate. Now, what is going to happen then when these two sides meet one another? Well, you're not just going to produce a single layered membrane here, okay? What you're actually going to produce is a double membrane, okay, like so. So the protein aggregate is actually going to get two separate phospholipid bilayers surrounding it. So here's the protein aggregate, and this is all resulting just from taking this vesicle structure and just moulding it around the protein aggregate and then fusing the two sides together. And of course this is a 2D picture, but if you can imagine it in 3D, moulding around there, it will form two spheres, two surfaces of spheres around the protein aggregate. Okay, now this structure that we've created here, this is now what's known as an autophagosome. Okay, so again, relating to the fact that this is going to participate in the autophagy process. So this is an autophagosome. Okay, right. What you're then going to do is bring a lysosome across. So let's say this is one of our lysosomes. So let's colour this in turquoise here. And you're going to fuse that lysosome with the inner membrane of the autophagosome. Okay, so let me show you this. So these two are going to now fuse together. Specifically, the inner membrane of the autophagosome is going to fuse with the membrane of the lysosome, and I'll bring this up a bit. Okay, so here's the inner membrane of the autophagosome fused with the membrane of the lysosome, and then the outer membrane will be like this. Okay, right. And then there is this protein aggregate now in the middle. Okay, and now we've got our lysosome fused onto there. Okay, now, the final thing then that you have produced here is what's known as an autolysosome. Okay, and the idea now is that these hydrolase enzymes are going to go and destroy this aggregate of proteins and remove it from the cytoplasm. So that's a good way of getting rid of protein aggregates if they do form. Okay, right. So that now completes our discussion of the proteostasis mechanisms. Okay, what we now want to turn our attention to is looking at the correlates of this in the form of the protein folding diseases, and we'll have a look at how protein folding can go wrong and how that can then lead to disease. Okay, right, so let me start off by saying that the diseases that we're going to talk about, these protein folding diseases, can be broadly divided into two categories. Okay, those where the disease phenotype results because of the loss of function of the protein. So, firstly, I suppose I probably should define what a protein folding disease is. A protein folding disease is a disease caused by a protein misfolding. Okay, so the loss of function protein folding diseases are the ones where the problems that stem from the problems that stem from this protein misfolding are to do with it no longer functioning. Okay, so this protein that has misfolded, it had some very important function. Now, once it's misfolded, it's no longer performing that function, and therefore that's creating all of the features of the disease. That's creating what's known as the disease phenotype. Okay, so the loss of function protein folding diseases are where the loss of function of the protein because it has misfolded is what causes the disease phenotype. Okay, and we're going to see two examples of loss of function protein folding diseases. Okay, the other category is then the gain of function protein folding diseases. And this is where generally the proteins that misfold, we don't actually know what their physiological functions actually are. Okay, so it doesn't seem as though them misfolding causes any huge deficit in physiological function. Okay, but instead, when they misfold, they generally start to aggregate now, and this is counted as a new function. They've gained a new function, and the aggregates that they form then are what cause the disease. Okay, so it's because the protein has misfolded and has now got this new function that you actually get the symptoms of the disease phenotype. Okay, so that's the broad classification of the protein folding diseases into these ones where the disease phenotype is caused by the loss of function of the protein that's misfolded, and the disease phenotype is caused by the misfolded protein now having a new function that it shouldn't have, and this is what causes the disease phenotype. Okay, so we are going to begin then with the loss of function protein folding diseases. Okay, so the first one that we're going to look at is an example of a lysosomal storage disease. Okay, so there are multiple different lysosomal storage diseases, but the one that we're going to look at is specifically called Fabry disease. Okay, so what happens then in all lysosomal storage diseases? We'll start by discussing the lysosomal storage diseases in general. Well, basically, in the lysosomal storage diseases, which by the way are often abbreviated down to the LSDs, okay, what happens is the proteins which misfold are the proteins that are within the lysosomes. Okay, so remember what lysosomes are. They're these cytoplasmic vesicles containing hydrolase enzymes. So in the lysosomal storage diseases, you get problems with the folding of those hydrolase enzymes, okay, within the lysosome. Okay, so what does that then cause? Well, the protein has misfolded. That's going to cause problems with its function. Okay, so the protein's function is lost, or at least reduced. And what does that then lead to? Well, what it leads to is the build-up of whatever molecule it was supposed to destroy. Okay, so remember the lysosomes are really this place within the cells where you send all sorts of molecules that are destined to be destroyed. Now, we've talked about them in the context of destroying proteins, but it's not just proteins. All sorts of other molecules are sent to the lysosomes to be destroyed as well. Okay, so if you have now lost function in a certain one of the enzymes in the lysosome, what it's going to lead to is whatever molecule that enzyme was supposed to be destroying is now going to build up within the cell. Okay, so let's show our enzyme here. This is the lysosome here, and whatever the substrate for that enzyme is, that's no longer going to be destroyed as efficiently if we've lost function in this enzyme, and therefore that's going to build up within the cell basically. And that's what underlies all of the lysosomal storage diseases. Okay, and as I say, there's loads of different lysosomal storage diseases, and what's different between them is that it's different members of the enzymes within the lysosome that are getting the problem basically. Okay, so let's discuss which enzyme is specifically going to have the problem in Fabry disease. Okay, so the enzyme that's going to have the problem in Fabry disease is what's known as alpha-galactosidase A. Okay, so alpha-galactosidase A, which for short is abbreviated to GL for galactosidase, and then A. Okay, so alpha-galactosidase A. Okay, now we'll see what the substrate for this is later on. What I firstly want to tell you about is the gene for alpha-galactosidase A, which chromosome it's located on, because it's an interesting answer, okay? And also the different types of mutations that can occur in alpha-galactosidase A. So, firstly, let's start off with which chromosome alpha-galactosidase A's gene is on. Okay, so basically, Fabry disease is an X-linked disease. Okay, it has X-linked inheritance. So that's because the alpha-galactosidase A gene is on the X chromosome. Now, you might be getting excited because X-linked diseases have that really interesting pattern of inheritance, okay, where boys are much more likely to get it than girls, but not in the case of Fabry disease. Okay, so let me just explain this a little bit more. So, basically, girls have 2 X chromosomes, okay, so I'll draw an X chromosome here, okay, and here is another X chromosome here. Okay, so if you're female, you have 2 X chromosomes, XX, whereas if you're male, you have 1 X chromosome and 1 Y chromosome, XY. Okay, now the Y chromosome does not have the gene for alpha-galactosidase A on it. So if you're male, you actually only have 1 copy of the gene for alpha-galactosidase A, whereas if you're female, you have 2 copies of the gene for alpha-galactosidase A. Now, usually, in X-linked diseases, it's the case that if it's a loss of function mutation, which indeed it is in this case, you're going to have some mutation in this gene for alpha-galactosidase which causes loss of function. It's usually the case that if you're male and you have a mutation for loss of function of this protein, then you get the disease. And then if you're female and you have 1 copy of your alpha-galactosidase A genes with this loss of function mutation, then you don't get the disease because you've still got 1 functional one over here. In the case of Fabry disease, you get it less severely if you're a girl, but you still get it even with only 1 gene with the mutated alpha-galactosidase A. Okay, so you don't need, in the case of a girl, to have both genes mutated. Okay, 1 is enough. Okay, it's because if you lose just 1 of them, the other one can't cope, even though it's still there. Okay, so girls do get a less severe form of Fabry disease than boys, because boys have completely lost function of the alpha-galactosidase A protein, but they do still get a disease phenotype, okay, it doesn't pass them by, okay, which is less exciting than for other X-linked diseases, where usually what happens is girls will only get it if they have 2 loss of function mutations in both of their copies of the alpha-galactosidase A gene, but it's not that case for Fabry disease. Okay, right, so let's now discuss the different types of mutations that can occur in alpha-galactosidase A. Okay, so there are many different types of mutations that can occur in alpha-galactosidase A. The most severe ones are where you either get a premature stop codon, which are known as truncations, or you get an insertion or a deletion, which collectively are known as indel mutations, that are out of phase, so an out of phase indel mutation. Okay, so let me explain what these 2 things mean. Okay, so let's just have a little bit of a reminder of how translation works. So if we have our mRNA here for the alpha-galactosidase A gene, then what's going to happen is the ribosome is going to work its way along the mRNA, and it's going to look at triplets of organic bases, okay? So there's a triplet here, and it will look at that triplet and decide upon which amino acid needs to be put in for that triplet. Okay, and a triplet of amino acids, of organic bases like this is known as a codon. Okay, so it will decide, looking at that codon, what amino acid should be put there. Then it will move on to the next codon here, and again it will decide which amino acid should be put there, etc., like so. Now, basically, the ribosome will go on all the way until it finally gets to a stop codon, which will be maybe over here, not necessarily right at the end, okay? And this stop codon will not have an amino acid that should be put in its place. Okay, that's the characteristic feature of stop codons. There is no amino acid that is put in for the stop codon, and instead the ribosome just stops there, and then the protein is finished. Okay, so all of the amino acids that have been added prior, that will be the polypeptide now. Okay, in truncation mutations, what occurs is you get some sort of mutation in the gene that then leads to a change in the sequence of organic bases in the mRNA, obviously, and then what happens is one of these codons earlier on up the mRNA will end up now being a stop codon. Okay, so maybe this one here. Okay, so you end up with what's known as a premature stop codon. Okay, and that will stop translation far too early. Okay, and the result of that is that you'll end up with a polypeptide that's much, much shorter. Okay, so if this is the normal polypeptide here, you might end up with a polypeptide like this, okay, that is correct for the first portion, but then is just stopped far too early. Okay, that's what is meant by a truncation mutation. Okay, so those are very severe mutations. Those result in no functional protein actually being produced. Okay, now what do I mean by indel out of phase? So indel is short for insertion or deletion. Okay, so you either insert new organic bases or you delete some organic bases. And we're specifically at the moment talking about insertions or deletions that result in a phase shift. Okay, so they're going to result in the mRNA being read out of phase. Okay, so let me try and explain this to you. Okay, let's say that we were to delete that organic base there in that mRNA. What would that now result in? As far as the ribosome is concerned, reading this mRNA. Well, now when it gets to this codon here, that one won't exist anymore. So now what's going to have happened is you're going to have effectively shifted all of the downstream mRNA up one. So this one here, this one here, and this one here is now going to be read as a codon. Okay, then after that, this one here, and this one here is going to be read as a codon, and then onwards, this one here, and whatever comes next will be read as a codon. Okay, so you've basically shifted the reading frame, as it's called, of the mRNA along by one. Okay, and this is what's known as a frame shift, because you have shifted the reading frame. Now these are dangerous because these codons might be completely different. In fact, they almost certainly will be completely different to the codons here. So you can end up making a protein that has a completely different sequence of amino acids, basically, within it. Okay, so this is what's known as a frame shift. Okay, and translation is occurring out of phase. Okay, so in that case, we deleted one organic base. Can you see that if you deleted two organic bases, the same thing would happen? You'd end up shifting the reading frame. Okay, however, if you were to delete 3 organic bases, you'd delete one codon, but you wouldn't shift the reading frame. So for instance, if I deleted all of those 3 there, you'd get rid of that codon, but then the codons after that would still be read in the same reading frame, and therefore that would be a less severe deletion, okay, and that would result in a much less severe change in the protein, basically. Okay, now, in addition to deleting organic bases, inserting organic bases can do the same thing. So let's say I insert a new organic base in, let's say, where should I put one? I'll put one in there. Okay, a new organic base in there, I'll colour this one in turquoise. That would also result in a frame shift, because now, when we do these 3, that would be one codon, but now the next codon will be this turquoise one, this one here, and this one here, and then the next codon will be this one here, and then the 2 after that, and then from then on the codon's reading frame would be shifted. Okay, so insertions can also shift the reading frame, and again, insertions of 1 or 2 organic bases are going to shift the reading frame, whereas insertions of 3 organic bases are not. They're just going to result in an additional amino acid being put in. Okay, so when I say indel that is out of phase, that's what I mean, that you are inserting or deleting a number of organic bases that is not a multiple of 3. Okay, not a multiple of 3, and this results in a frame shift. Okay, not a multiple of 3. Now those are very, very serious mutations that really are going to result in absolutely no functional protein being produced from that gene. Okay, right. Less severe mutations are indel mutations that are in phase, so in phase indel, where you insert or delete a number of organic bases that is a multiple of 3. Now obviously this needs to be taken within reason. If you insert, let's say, a sequence of organic bases that is a multiple of 3, but is 900 organic bases long, then that obviously is going to count as a major mutation. But if we're, say, just inserting in 3 organic bases, that will be counted as one of these more mild mutations. Okay, right, so I should probably just put small as well. In phase, multiple of 3. Okay. Right, and then the other type of mild mutation that can occur is what's known as a missense mutation. Okay, now a missense mutation is where you literally substitute one organic base in the mRNA for another. Okay, so maybe we change this one here for one different organic base, and now that means that this codon is different. And then the key thing is in the missense mutation that codon is now going to code for a different amino acid. Now that's not trivial, okay, because you can change an organic base within a codon and it can still code for the same amino acid because of the degenerative nature of the genetic code, the fact that there are more codons, 64 in fact, than there are amino acids, 20. Okay, now 3 of the codons are used as stop codons, but that still leaves 61 possible codons for just 20 amino acids. So many codons code for the same amino acid, so some amino acids have more than one codon coding for them. So it is plausible that you could change an organic base and it not actually change the amino acid, but a missense mutation is where you change the organic base and it does result in a change in the amino acid there. But all that does to the protein is it changes one amino acid in the protein. All you do is take one amino acid out and put a different one in there. Okay, so that again is quite a mild mutation. Okay, so it is these ones that we are interested in. These are the actual protein folding ones. Okay, these are the ones where the disease is caused by a protein folding abnormality. These ones are completely different. This has nothing to do with protein folding in this case. Okay, these are never going to produce functional proteins, no matter how good your protein folding machinery is. Okay, so we've got these more mild mutations here. There are 2 options for how these more mild mutations can then lead to problems in the alpha-galactosidase. Either the alpha-galactosidase now misfolds, okay? Well, actually, that's for sure, it misfolds. And then there are 2 options. Either it's allowed to live, i.e. it's not destroyed by the cell's degradation mechanisms that we've discussed, the ubiquitin proteasome pathway and autophagy, okay, but it's dysfunctional, okay, so that's one option. It can misfold and then it's dysfunctional, or it can misfold and it's still functional, but it gets picked up by the cell's degradation mechanisms and destroyed because it's a misfolded protein. Okay, so it's degraded by the ubiquitin proteasome pathway or autophagy. Okay, so these are the mutations that we're interested in with regards to looking at these lysosomal storage diseases from a protein folding standpoint. And the reason that they cause problems is they lead to the misfolding of the protein, and now either it's allowed to survive but then is dysfunctional and doesn't perform its role anymore, doesn't catalyze the reaction that it catalyzes, or it's actually degraded by the cell's quality control mechanisms, so for instance the ubiquitin proteasome pathway or autophagy. Okay, so let's now actually discuss then, what does alpha-galactosidase actually do? Okay, well, it breaks down a molecule known as globotriaryl ceramide, okay, which is a great mouthful, so let me write this down. So, globotriaryl ceramide. Okay, right, now this is a lipid molecule, in case you're not familiar with what globotriaryl ceramide actually is. Okay, so let me draw a little picture out to try and explain what it actually is. Okay, so the first clue is this word ceramide. Now, ceramides are a massive great family of lipid molecules that are all based on the molecule sphingosine. So let me draw a cartoon for this. So this line here, which I will colour in orange, this is going to represent a molecule called sphingosine, okay, which is the backbone for all ceramides. So this can represent a molecule called sphingosine, and it is a lipid molecule. It has a very, very long tail here, okay, that is very, very hydrophobic, very, very neutral. Okay, now at the head it has some more interesting functional groups. So for instance, on its first ever carbon, okay, and it's actually an 18 carbon molecule, on its first carbon it has an alcohol group, and on its second carbon, if memory serves right, it's got an amino group. Okay, it is an amino group off the second carbon, and off the third carbon it also has another alcohol group. So there are some interesting groups right at the start here, and then the rest of it's just extremely hydrophobic, okay, and very lipidy. Okay, now, to make a ceramide, what you do is you add some long-chain carboxylic acid attached to the amino group off the second carbon here. Okay, so that's some long-chain carboxylic acid. And this molecule that you've now created, that's what's known as a ceramide. Okay, so it's not just one molecule, because we didn't state which specific long-chain carboxylic acid we were going to add on here. Okay, so there were loads of long-chain carboxylic acids that you could add on there, and they'd all be considered ceramides. So this is some long-chain carboxylic acid, or the other name that biochemists like for long-chain carboxylic acids is fatty acids. Okay, so this is a fatty acid rather. Okay, right, now the globotriaryl. Okay, well this refers to a sugar group that you are going to add on to the alcohol group that comes off the first carbon of the sphingosine molecule here. Okay, and in fact you're going to add a little trisaccharide molecule here. Okay, so I'll just draw little hexagons to represent monosaccharides here. So you add on 3 little monosaccharides here in the trisaccharide, and this then is the finished globotriaryl ceramide molecule. Okay, so you've got these 3 saccharide molecules, 3 sugar molecules attached on the head there, and then the lipid groups back here. Okay, so that's the structure of globotriaryl ceramide, and it's a normal lipid that is found within all cells of your body. Okay, now, it is the job of alpha-galactosidase A to break down the globotriaryl ceramide. Okay, now, we are saying that we have got a mutation in one of our genes for alpha-galactosidase A. Whether we're a boy or a girl, this is going to cause loss of function of alpha-galactosidase A. Okay, either because it's a really severe mutation and has resulted in complete loss of synthesis of any functional protein that bears any similarity at all to the original protein, or because it's one of these less severe proteins and then it's failed to fold properly, so it's a true protein folding disease, and then it's either dysfunctional because it's folded incorrectly, or it's been degraded because it's folded incorrectly. Okay, now, as I was saying earlier, even if you are a girl and you still have one functional gene for alpha-galactosidase A, the loss of function in one of those copies has now resulted in too little alpha-galactosidase A function for you to not actually get some symptoms of Fabry disease. As I said, you get it less severe than a boy would get it. Okay, but you do get symptoms. Okay, so what then are the symptoms of this? Well, the problem is that alpha-galactosidase A lack of function is going to lead to this molecule, globotriazosyl ceramide, building up now in all of the cells of your body, and this is what then leads to the symptoms of the disease. This is what leads to the disease phenotype. Okay, so let's discuss what the symptoms of Fabry disease actually are then. Okay, so one of the key symptoms then of Fabry disease is what's known as acroparesthesia. Okay, now what does this mean? Acroparesthesia means that you get episodes of pain in your limbs. Okay, so paresthesia refers to tingling or painful sensations in particularly your fingers and your hands and your feet as well. Okay, so the extremities. Okay, paresthesia really refers to like pins and needles experience that we're all used to. Okay, now acroparesthesia particularly means episodes of pain in the extremities, so in the hands and the feet. Okay, so episodes of pain in the hands and the feet. Episodes of pain in hands and feet. Right. Now, that's one of the key noticeable symptoms for people suffering with this, okay, but the more severe symptoms that occur are that it can lead to eventually problems with the kidneys, okay, so it can lead to kidney damage and then eventually kidney failure, okay. It can also lead to vascular disease, okay. And the reason for this isn't really well understood, okay? Initially, it was thought that the reason that it led to vascular disease is that it would accelerate atherosclerosis in some way, okay? But it doesn't seem to actually be accelerating atherosclerosis in people with Fabry disease, i.e. if you look for signs of atherosclerotic plaques in these individuals, you don't seem to find them. Nevertheless, they get vascular disease, okay, so they're at huge risk of getting a heart attack, myocardial infarction, and also suffering a stroke, okay, but it doesn't seem to be a mechanism that's driven by accelerated atherosclerosis, which was what people first assumed that would be happening because of the increased level of the globotriazosyl ceramide. Okay, right. So that now concludes our discussion of Fabry disease, which is the example we're going to give of a lysosomal storage disease. Okay, and that was caused, at least in some people, by mutations that led to misfolding of the protein for alpha-galactosidase A. Okay, in the next video what we will turn our attention onto is cystic fibrosis.